Opportunistic yeast. So we're going to talk about Candida albicans and Cryptococcus neoformans. For yeast, they are normal flora, oftentimes in the sputum, urine, feces, and vagina. They keep the bacterial levels in check, so to make sure there's a healthy balance. However, yeast can be pathogenic, so if they are recovered from what is normally considered a sterile body fluid or tissue, or they're present in pure culture or in large numbers from any culture site, they are going to be considered pathogenic at that point. They are budding organisms, and growth on agar will be colonies that are smooth and have a creamy looking texture to them. So Candida, this is normal flora of the gastrointestinal tract and the mucocutaneous area of the oral cavity. It will cause thrush if the balance is off in the mouth, and women do have vaginal yeast infections, so it's called manila or vaginitis. We do need antimicrobial agents that will inhibit the normal flora, therefore the yeast can become pathogenic. So some women, if they're taking an antibiotic for a bacterial infection, it can wipe out enough of the normal flora in the vaginal tract, and then it results in a yeast infection because the yeast kind of takes over instead of the typical stuff. Hormonal imbalances or use of antimicrobial agents associated with those will sometimes result in superficial candidiasis. But yeast infections can also disseminate, can travel throughout the body. So these are associated with a defect in the function of phagocytic cells or cell-mediated immunity, such as AIDS. Cryptococcus is caused by Cryptococcus neoformans, and this is a multi-system disease. The portal of entry is typically through the lung. There are 6 clinical types, which include pulmonary, central nervous system, visceral, osteoporosis, cutaneous, and mucocutaneous. So the infection is first pulmonary, and then it can disseminate to those other systems. It could be benign, and it could be acute. With Cryptococcus, primary pulmonary can be asymptomatic in the general population. We'll see central nervous system Cryptococcus infections typically in the acute settings, and this results in meningitis or brain abscesses as well. Pigeon excreta is the chief source for distribution and maintenance of this organism. This has a capsule, therefore it's protected from phagocytosis. Serotype A is the most common type from a clinical infection standpoint that you will find. Identification of both of these yeasts, they're usually recovered from sputum, urine, or cerebrospinal fluid. Sputum and urine, you will just set up a normal bacterial culture. For the spinal fluid, you will actually concentrate that specimen by centrifugation. You will then use the concentrated or the sediment to direct plate onto media as well as a gram stain. Cryptococcus neoformans, you can do that on an India ink stain as well, but that's not routinely done on every spinal fluid. That's only if you have a physician order, they have like a suspicion as to what this is. There are also latex agglutination kits you can use, or you could do a direct exam of your specimens with a KOH. Cryptococcus species produce brown pigmented colonies when grown on media containing caffeic acid due to the production of melanin, which is a useful diagnostic feature. DNA probes are available for the rapid and specific identification of Cryptococcus neoformans as well as Candida albicans. The germ tube test is commonly used to identify Candida albicans as it produces characteristic germ tubes within hours of incubation. Additionally, carbohydrate assimilation tests, such as the API-20C system, are used to differentiate and identify yeast species based on their ability to utilize various carbohydrates.